Cross4Rare gives access to a comprehensible research opportunity and strategy service. In this program, we can provide you with research status, gap identification, how to attract pharmaceutical company to your disease, and develop your scientific community. We give you ways to support drug development, from target identification to clinical trials, as well as a target scan service. The goal of most foundations are to cure or find treatments and improve patients' daily life. The purpose of this presentation is to help foundations understand the different steps and requirements necessary to transition to a drug development support. To be active and find a cure, foundations need to be involved in drug development, and at a certain stage in the process, will need the intervention of a pharmaceutical company. We will guide you on how to be prepared to convince private companies to invest in your disease. The first step is to understand the process to get to a treatment or a cure, how to develop and bring a drug to the market, who the stakeholders are, when to interact with the different stakeholders. The road to a drug on the market is often represented by a horizontal flowchart. Starting from investigation to characterize the disease, then molecules, also called chemical compounds, are tested for the disease, undergo modification for optimization and safety to become a potential drug. These different steps are called the preclinical development. When the optimized chemical compound fulfills the requirement to be tested in human, the clinical phases are performed before going to the market. Where are the different steps performed and by whom? Research is executed in scientific academic centers, mainly universities and NIH, and is focused on the biological characteristics of the disease, so performed by biologists. The first step, really concerning drug, is when you enter the preclinical studies. It is performed in either biotechs, pharmaceutical companies, CROs, or specialized governmental organizations such as NCATs, and sometimes in academic labs. For successful preclinical studies, you need to have pharmacologists, who are biologists specialized in pharmacology, working together with medicinal chemists and admit toxicologists. The clinical phase are performed in hospitals by clinicians. The drug is brought to the market by a pharmaceutical company with the business development necessary. The process is regulated by legal, financial, and human aspects. The patient, through the foundations and patient advocacy groups, need to be very actively involved in the whole process. They are the support and beneficiaries and should be at the center of the process. The FDA should not be reduced as the judge at the end of the process. They are a tremendous support all along the drug development. They will help you address the right issues from the beginning, as I will explain in this presentation. And of course, the pharmaceutical and biotech companies are the major power in the drug development process and manufacturing. Until recently, the technical process of drug discovery was exclusively owned by pharmaceutical companies in terms of knowledge, expertise, money, and decision-making. For a few years now, the foundations became proactive and involved in pharmacology, pushing or attracting companies to their fields and their disease. The problem is that the knowledge in pharma is kept in pharma, and if you were not trained in the industry, there are no school and academic labs that will give you this expertise. Foundations encounter diverse challenges that a report from the Milken Institute nicely exposed. This is a table from their report. It is noticeable that the key challenges are what makes the daily job in the pharmaceutical industry. Validation and translation study is the starting point of any project.
then followed by the drug development in collaboration with medicinal chemist groups. Foundations need the right partner at the right time, but they don't really know who and when. And they have limited knowledge of business strategy and development. There is also the challenge of IP. They don't have easy access to patent attorney as the private sector or university do.
The action points foundation can take to tackle these challenges will vary among the different type of drug discovery process. Treatments can be separated in two large groups: the small molecules, which are the traditional chemical drugs, and the biological-based treatments, highly innovative and for a part of them still experimental. We will go through the most representative to see where to fulfill the gaps and tackle the challenges. Through the traditional target-based drug discovery process, the repurposing drug, the phenotypic high-content screening, and also through the cell and gene therapy, which have their own challenges. What is extremely important is not to be exclusively focused on one type. The secret for success is to balance your interest.
You need to have a balanced portfolio between the risk of failure in development and the benefits your patient will get from the treatment, starting from symptomatic drugs that you have already with low risk but low reward, disease-modifying therapy with repurposing drugs, so you increase the risk a little bit, but the reward is still not really high because they are not very specific to your disease. The new molecule entities or disease-modifying therapies that are at higher risk but highly rewarding, and at the top, of course, the gene therapy.
The first process I will explain is the traditional target-based drug discovery process. It starts with target identification and validation, where you choose on what you are going to work and be sure that it will improve the condition of the patient. These steps are performed by biologists. Then, medicinal chemists work in collaboration with the biologist to test molecules, improve the molecules, and test them in animals. When a molecule is proven active, it is also necessary to check it for safety and finally evaluate the toxicity and the efficacy of the drug in humans before reaching the market. This process takes more than 10 years and costs millions of dollars. The first step is target identification and then validation. Target identification is where you can intervene to repair the broken path. There are two levels of intervention. First, tackling the origin of a problem, which in most rare diseases is a gene mutation. The target would be the gene or the gene transcript. Second level is the consequence of the problem, the direct consequence. The protein resulting from the gene mutation does not exert its function in the cell. The target would be the protein itself with protein replacement. The indirect consequence, what I call the co-workers, are the elements not getting their information from the protein to be activated or deactivated. The target can be any downstream signaling pathway. After having identified the target, it needs to be validated. That means checking if modifying the target activity will change the manifestation of the disease. At the gene level, validation is performed by engineering the gene back in the disease model and assess if the syndrome or disease characteristic are decreased or reversed. The same idea is followed for the transcript, yes or no answer, depending on the limit criteria to validate the target. The protein replacement in the model will be assessed the same way to be validated. For the downstream signaling pathway, the inhibition or activation of the target by existing chemical compound in several models of the disease will reveal if the pathway is relevant to the disease. Now that you have a target, you need to find a molecule, a future drug, that is able to activate or block it depending on its function in your disease. Testing a lot of molecules is called screening. You will identify hits that become leads if the hit has a high effect with a low dose or concentration. To perform a screening, you need tools. An assay, which is how to evaluate the action of a molecule on the target, and test systems, cellular models and animal models. There are different types of source of molecules. What we call the traditional drug discovery new molecule entities with the traditional drug discovery pathway. Then you will see the phenotypic drug discovery new molecule entities or the repurposing drug. To find a new molecule entity, also called a compound, the screening is performed with chemical libraries. The compounds are incubated with the target. The compound binding or altering the function of the target is called a hit. Based on that hit, the chemist will find a family of compounds. They will modify their chemical structure and test every modification to make a kind of map that will associate the different part of the compound to its activity and characteristics, and by that way optimize the lead compound for the best efficiency. It is called establishing a structure-activity relationship, or SAR. Where can you perform screening? Either in biotechs, CROs, or less often now, pharmaceutical companies. The public option can be establishing a collaboration with the NIH-affiliated center specialized in screening with medicinal chemists and all the tools you can dream of. It's named NCATS, for the National Center for Advancing Translational Science. Having a meeting with them will help you establish the details and plan for a screen. Also taking into account all the material and knowledge you have on your disease. You are also free to perform your screening in academic labs, but keep in mind that they will not have medicinal chemists and highly specialized pharmacologists, so you will need to seek for this expertise externally. The choice and access to chemical libraries are also separating between private and public, but also between general or specialized libraries, such as the MIPE, available at the NCATS, that will have compounds specific for certain disease areas. In every single type of treatment, disease models are the cornerstones of the process. From validating a target to screening, testing the efficacy of a drug, or assessing the toxicology, models are necessary. The cellular level models are more commonly developed, require less investments and less specialized knowledge and training than animal models. Cell models are usually used in large-scale experiments, such as screening. Here, on the left, is presented the high-throughput content with thousands of possibilities to assess and test in the same experiment. At the opposite, on the right side, is a low throughput, with one compound in one animal. Among cellular models, the throughputs can be different if the model is based on animal cells or on patient-derived cells. The complexity increases as you reach the phenotypic model based on iPSC and even human iPSC. Animal models vary as well on the level of throughput, from non-vertebrate to different vertebrate models with non-mammals, mammal rodent, and at the end, non-rodent mammal. The choice of the model will depend on the kind of drug or treatment that are developed. The challenge is that it's time and money consuming to develop and maintain an animal model. So every research team who develop one will tell you that their model is the best and will often ask a lot in the IP negotiation to use it. Having access to the model might be one of your great challenges. An alternative to the target-based drug discovery process is the repurposing drugs. In that case, you take a drug that is already on the market, so already went through the whole process. Test it directly in your cellular model or even directly in the animal models. If you see disease modification, you can go directly to human testing, skipping the toxicology and ADME tests, since the drug is already on the market. It tremendously reduces time and cost, but will be less specific to your disease than the drug that went through the traditional process. A third type of discovery process is the phenotypic high-content screening. It is an efficient strategy when you don't know the mechanisms of action, but you know the end manifestation of the syndrome and have genetically modified models representing the disease. Let me explain briefly the technique. At the beginning of the development, the pre-embryonic stage, there is just a mass of cells that all look similar. That is, here, for example, a stage of blastocysts, you cannot see an arm or the head. These cells are stem cells, and they will undergo a process of cell differentiation to become more and more different, as, for example, neurons are different from muscles. Recent discoveries in embryonic developments identified the factors that can reverse the differentiation and go back to the stage of stem cells. How is it used? The patient cells contain their genetic material, therefore their mutation. We can induce the cells to become pluripotent stem cells and then differentiate them in the relevant type of cells for your disease. Here is the example of skin cells becoming at the end cardiac cells. Then we can use another technique extremely useful. The cardiomyocyte can be grown in such condition that it will become a 2D functional model, contracting cardiomyocytes, showing all the characteristics of the patient disease. The phenotype of the patient is maintained and can be studied under different drugs or conditions. You can measure the contraction force, the electrophysiology, the metabolic activity, or observe the morphology of the cell. The remarkable part is the high-content screening. Because this model is based on cell culture and not animals or tissue, you can have as many cells as you want, so the scale of sample can be really high. The experiments are realized with cells coated on plates the size of your hand that will contain 96 small wells in which you can realize a different condition in each well. So you can test, for example, 96 drugs at the same time. The flowchart is shorter than the two previous drug discovery models. You start from screening, but you can measure several aspects at the same time, including some toxicology. So those effects saves a lot of time. In fact, phenotypic screening has been a dream for years as an alternative to target-based drug discovery that in some cases has its limits, such as poor understanding of the disease and complex causative mechanisms, leading in fact to expensive late-stage failure, which of course challenges financial viability for pharmaceutical industries. Phenotypic screens measure more complex mechanisms, so closer to the disease. They became possible thanks to the iPSC, which is induced pluripotent stem cells for humans, and the genome engineering combining those two, and thanks to big data analysis and small molecule library improvement, and now even more promising with the 3D cell culture and organoid models. In the biological-based therapy, I will focus on gene therapy. The perspective of having a cure is everyone's dream. Unfortunately, gene therapy is not a possible option for every disease. It is still at the beginning of the technical investigations, but up to now, only monogenic disease can use gene therapy, and the size of the gene is a limiting factor. The different type of gene-based therapy are first gene replacement therapy. It replaces the whole gene by adding copies of the gene in the nucleus of the patients. That is the ideal solution. Gene editing is when you add, remove, or replace a piece of the patient's gene. It is still experimental. There is no FDA approval yet. The common technique is the cluster regularly interspaced short palindromic repeats, or CRISPR. Many other editing innovative techniques are being developed that will help the disease that cannot use gene replacement therapy. Last, I will talk about is the patient-derived cellular gene therapy products, or CAR-T cells, that stands for chimeric antigen receptor T cells therapy. The T cells, a type of immune cells from the patient, are collected. A specific gene is added to the cells in order to recognize and destroy specific cancer cells. The cells are produced in high quantity and infused back to the patient. So first, organize a workshop with key opinion leaders to determine which type biological-based therapy, if any, is possible and best adapted to your disease. The different gene therapies can be separated into two big groups according to its way of delivery to the patients, the ex vivo and the in vivo therapy. The in vivo has the biological product or the gene in a vector directly infused in the patient, while the ex vivo has patient cells collected, then genetically modified, produced in high quantity, and introduced back into the patient. For both systems, the cost, the time, and the quality are challenges. The cost is high because of the necessity to work in GMP clean room. The number of full-time employees is high, and the manufacturing is not yet at an industrial level. It is time-consuming because there is almost no automation, the purification techniques are too slow, and there is a shortage in vector supply. The quality is more demanding than in traditional therapy for safety reasons, and there is a lack of stability of the vector. The number of deviations per batch is too high. So how can foundations help in the process? The main roadblock in the process comes from the fact that in the rush to get to the market, scientists did not lock down the biology. There is a lack of understanding of the biology for safety and industrialization process. I will not go further deep in explaining the techniques. There are very nice webinars organized by Nord. You can refer to them if you need to better understand the biology of gene therapy. Here is where the intervention should be. For the biotech and pharmaceutical companies, the highest priority is manufacturing, how to go to industrialization. For the foundations, the list of action points is long, since the basic science has been skipped and the field really suffers from it now. Foundations should be investing in innovation studies for new technologies, for example, adherent versus non-adherent cells, investigate stabilization in the genome, in ways to improve the yield. Recovering empty cassette is a lot of loss of time and happens a lot of time also. They should investigate in cell health, pre-existing immunity and elicited immunity to AAV vector. For the regimens, since the AAV is not integrated in the genome, there is a high probability of a redosing. So investigate the use of immunosuppressive for redose. Invest in high-risk technologies. training of technicians, which is really a lack now. They lack people knowing the techniques. This list is not exhaustive, and your foundation should prioritize and tackle the most relevant points for your disease. The FDA is the inevitable partner. They developed several programs in support to gene and cell-based therapy, the RMAT and the Interact programs. I invite you to look at them. In addition, Peter Marks presented the project of a consortium to help reduce the time and increase the quality of gene therapy process. He made a comparison between the process as it is now, taking 4 to 8 years, with the process as it will be in the consortium, taking 2 to 4 years. Both systems initiate with the investigator having the therapeutic concept, but then the development of the construct will be guided by the consortium and given to their GMP facility for them to produce a GMP material for preclinical studies. There is a standardization of quality and protocols satisfying the FDA requirements, so that you create and submit an IND, investigational new drug, much faster. Therefore, get to the patient much faster. The consortium is one option among others. If your project of gene therapy is already advanced and the viral vector construct is developed, the option to go further in the production with the GMP facility are first to go to CDMOs, contract development and manufacturing organization. There are more than 50 and new ones every week. I listed here a few of them. There is also the option of a collaborative facility where you can access and utilize labs and expertise. For example, the Cell and Gene Therapy Development Center at the New Jersey Innovation Institute. That will give you access to GMP suites and all capabilities of running clinical trials. You will have the possibility to train in the techniques of gene therapy, and you will be in contact with technology developers, process developers, and biotech and industry. A third option is the FDA consortium, as I mentioned before, that makes technology more accessible to academic innovators, streamline preclinical and manufacturing information needed for investigation and licensure, facilitate the development of manufacturing and other advances that will ultimately benefit the entire fields and increase value to asset both investigators and industry. We went over the main type of therapies up to now. To summarize the action points foundation need to take in the preclinical part of their portfolio, I took back the target-based process as reference. What we have seen along the flowchart is create a balanced target portfolio, define target product profile that, we didn't see it, I will explain it shortly after, take into consideration repurposing drug testing. If your disease has a complex phenotypic profile, favor the phenotypic screening. And is gene therapy for your disease and where to invest? Be ready with the animal models, taking into account that most of the time, but not always, the FDA needs 2 mammal models, including 1 non-rodent, preferentially big mammals. Why do I make a pause here? Foundation for Rare Diseases are the engine that makes the drug investigation going further. Even if the foundation invests in different stages of drug development, there will most certainly be the need for pharmaceutical companies to take over, either in screening, providing chemical libraries, or later at the most expensive stage, the clinical developments and clinical trials. To convince the pharmaceutical companies to join and invest in your project, you need to de-risk the project. What makes the risk for pharmaceutical company is too high investment in time and money, project failure at early stage, and of course, FDA not approving the drug. So how can we tackle that? When you contact pharmaceutical companies or biotech, they will ask questions to evaluate the risk and benefits they will take to get involved in your field. If you can demonstrate that you have developed a de-risking pharma platform, as they have done at the Children's Tumor Foundation, it will be much easier to get them on board. They can ask you, where are the patients? You need a patient registry, which is important for the clinical trials, but also for the post-marketing surveillance. What about the patient engagement? You need patients as advisors and reviewers. What about the path to approval? FDA endpoints. It will be a global effort with clinicians, patients, and FDA, of course. I will talk about the target product profile and the standardized endpoint for clinical trials, meaning every clinician will measure the same aspect with the same precise methodology. They will ask, are there hospitals and clinicians trained and ready to perform clinical trial? Start working as soon as possible to build a clinic network. Do we have access to tissue? It would be nice if you had a biobank. I know it's expensive, but you can maybe group with other foundations to share the expenses. Do you have a variety of targets? Are they validated? That will be the role of your science director, or you can outsource this service. Another question will be, can we test drug easily? You need a variety of cellular models and animal models. Don't forget that you will need to get good IP agreements to be able to access the models. Start working on it as soon as possible. Do you have key opinion leaders? Organize conferences and workshops. And finally, it would be nice if you had a market model. When to prepare? The process seems to be linear over time, and some make the error to take it that way, losing a lot of time and efficiency, which is not the best way to convince a company to invest in you. Prepare all the tools necessary for each step before you get to that step. For example, if nothing is ready to measure clinical outcomes, the FDA won't let you put your patient at risk by trying a new drug, and the pharmaceutical companies know it. You need to come to them with at least a partial completion of the requirements. This takes years. Instead of first testing molecules in model and when an interesting molecule is found, starting to think about the next step and realize it will take years, start preparing the clinical stages before or at the same time as you start the academic science. The advice from the FDA are for starting points. The most important thing is to start with a consensus definition of the disease. This is fundamental before anything else. You can also establish diagnostic criteria and care guidelines. For that, organize clinicians' workshop. Take the occasion to contact and gather clinician specialists of your disease from different continents if you can. That will help increase the patient volume and stimulate the attention and the wish to help for your disease. The second point is define what your specific rare disease needs now. Most rare diseases have large and diverse phenotypes. Your patients are the expert in their disease. They will give the information for drug development and evaluation. The FDA advises to define the critical issues, the most important symptoms for the patients in their daily lives. It will facilitate the decision to approve a drug if it is proven that the target of the drug will be a benefit and change the patient's life. As an indication to what to look for, you can follow the guidelines from the FDA. I indicated the website page here. Mainly, identify the symptoms and impact of the disease on daily life, the current treatment options, the important disease subpopulation, or patient characteristics that should be represented, the phenotypic and severity variation, and if there is a need to focus on any particular subpopulation, such as children, for example. How to get all this information? The ideal would be to perform an externally led patient-focused drug development, PFDD, with the FDA help. An acceptable option is to gather as many patient, caregivers, clinicians as you can and follow the guidelines. Take into consideration that teenagers have a tendency to lower the intensity or importance of their symptoms. The patient-reported outcomes, what we call the PROs, have to be carefully evaluated.
In addition to a balanced portfolio, one of the primary steps to ease the path to pharma and increase the chance of success for treatment is to have a target product profile. This is established with a tight collaboration between the patients, the clinicians, and the help of the FDA. You need to define your goal at two levels: the ideal and the acceptable requirements for the compounds. You need to define primary product indication, the patient populations, the treatment duration, delivery modes, the dosage form, the regimen, the efficacy you want, and establish the risk and the side effect that you will tolerate, and try to find the ideal therapeutic modality.
There are additional benefits of establishing the patient needs. The area of focus will be defined easily. This will allow you to contact appropriate center of excellence in areas that might not have been your priorities, and develop appropriate cellular and animal models. On the drug development point of view, it will guide your choice of specialized biotech or pharma to partner with for drug development or repurposing drugs. But it will also guide the choice of specific compound libraries. You might see areas also to expand and an opportunity for your patients to define as a subgroup to join clinical trials from other diseases. Going back to the flowchart, what are the points to prepare for the clinical phases of drug development? First, keep improving the natural history by improving your registry. Create a clinical network, define FDA endpoints and criteria, and mostly for ultra-rare disease, increase patient volume. One of the most important tools for the clinical phase is the patient registry, because many other points are depending on it, as you will see. A registry is a collection of standardized information about a group of patients who share a condition or experience. The common benefit of the registry is that it increases the knowledge about the disease, but it is much more than that for each stakeholder. For the patient, it creates a community, facilitates the development of new treatments, and increases access to care. For the patient advocacy group or foundation, it is to support training and research in disease area, and to raise the profile of the disease to encourage funding for more research. The clinicians and principal investigators learn from the registry community, gather data to refine complex or undefined diagnoses, but also develop and inform treatment guidelines to target product profile and standard of care. Most of all, for pharmaceutical companies, it will allow them to understand the natural history of the disease, therefore design better clinical trials, evaluate potential relevant clinical endpoints, fulfill the post-marketing commitment, determine potential market share, access patients, and to a lesser point, generate publications. For the FDA, they can check the validity of the clinical endpoints. They can monitor the safety of approved products. evaluate cost effectiveness and budget impact, evaluate evidence for reimbursement. There are three types of registry. All can be national or international. The patient self-reported, where only patients are asked to provide information, that's the PROs. The professionally reported, where only doctors provide information. And the combination of patient self-report and professional report, where some information is provided by both patients and their doctors. The challenges you can encounter are to get the different clinics to agree to share the patient's data and to guarantee a quality and reliability of data entries. Do not try to do it alone. There are organizations that will guide you through the process. NORD, for example, has the I Am Rare program, and if you are interested in an international registry for Europe, contact Eurordis. The governmental organizations such as the FDA will be of good help with the CEDAR, and the NIH have the NCATS natural study initiatives. The private sector should be sponsors because it is quite expensive, and I put some vendors here available. I listed them, 6 of them. The next tool needed for the clinical phase is the clinic network. What is the purpose of a clinic network? First is to help patients find specialists. You can build a Find a Doctor map, for example, on your website. Develop access to centers of excellence all over the country. Standardize quality of care. Facilitate patient recruitment for clinical trials and increase awareness among the physicians. How to develop the clinic network? First, identify expert clinicians in your disease. Build a clinical community. Promote leadership. Fund and provide training for physicians. Create a clinical care advisory board. And finally, implement standard of care. This, of course, has a cost. Support stipends between $5,000 to $10,000 per center and per year. The choice of endpoint is extremely important in clinical trials. An endpoint is a primary outcome that is being measured by a clinical trial. A clinical trial might use a clinical endpoint or a surrogate endpoint, which is predict clinical benefits such as biomarkers, for example. Ideally, surrogates are established or validated, which means that they are proven to predict clinical benefits. However, for rare disease, surrogates that have not been validated, but rather reasonably likely to predict clinical benefit, might be accepted in basis of the FDA's accelerated approval pathway. According to the FDA, endpoints are not necessarily what key opinion leaders or clinicians usually measure. It should be the points that matter for the patient. When to define endpoints? As soon as you can, because it takes years. How to define endpoints? You should do it during the patient-focused drug development, the PFDD, and also from natural history, retrospective, prospective, and longitudinal. If a natural history is not available in your disease, gather key opinion leaders and as much patients as you can, and identify a battery of outcomes and measures. Repeat that after 18 months and compare the results and determine the points of progressions. Try to develop a broad spectrum of endpoints. Find one that is clinically meaningful. The other endpoints will serve to complement that endpoint, because it is difficult to predict which one will work in a patient. To ease the process, remember that you can borrow clinical endpoints that have been developed for other diseases and attune them for your patients. For example, neurological measures, the 6 minutes walk test, Rare diseases have complex phenotypes, but some overlaps and are common to several diseases. To enrich patient population, I can see three levels of battle. The first one is an indirect way to increase by expanding the field and benefit from other disease. As I mentioned before, by determining what your patient needs, you might discover new areas of focus, and defining the endpoints of clinical trials, you might identify overlaps with other rare diseases with complex phenotypes. You can define and insert your patient as a subgroup in other clinical trials from cardiology, hematology, myopathy, neuromuscular oncology, or others that you would have to identify talking with your clinicians. The second way of enriching the patient population is by decreasing misdiagnosis and increase medical awareness. One way is to define the standard of care for each specific medical area in a clinician workshops and having poster and oral presentation in specific conferences where you will reach as many clinicians as possible in the specific areas mentioned before in the expansion of your field. The third level is the globalization of your foundation. CORD is the counterpart of NORD, the Chinese Organization for Rare Disease and the Chinese Rare Disease Research Consortium, the CRDRC, are relatively new organizations. They have funds for establishing the National Registry for Rare Disease, and they plan to identify 5 to 30 rare disease genes per year, implement molecular diagnosis and translational research based on these genes, and facilitate the development of therapeutic strategies for rare diseases. In Europe, you can get into Orphanet or Eurordis to create affiliates of your foundations or get in contact with European Association in the expanding field. This concludes our presentation. If you need any more information, you can contact us at www.arthosforkids.org or email us at armelpindon@gmail.com. Thank you for your attention.